Dear students, Assalamu alaikum. Today I am going to deliver a lesson for class 11th subject Zoology. Since our previous topic we have discussed about the cell division, during which we discussed in detail about mitosis, which is a common type of cell division that occurs in the somatic cells. Now today we will discuss about a special type of cell division called meiosis, which occurs in the gonads or the germ cells and is employed for the production of haploid gametes or germ cells. Now meiosis, the term meiosis was first of all introduced by Farmer and Mori in 1905. Meiosis is a cell division in which the number of chromosomes become half in daughter nuclei as compared to the parent nuclei. Thus it is also known as reductional division. Now associated with the formation of germ cells or gametes. Meiosis involves two sequential cycles of nuclear and cytoplasmic divisions, which are known as Meiosis-1 and Meiosis-2. Now during this meiotic type of cell division, the parent diploid cell, which has a diploid number of chromosomes, actually undergoes divisions at two stages. At the stage first, that is the Meiosis-1, there occurs reductional division, which results into two cells with haploid number of cells. Then these cells, which are produced as a result of meiosis-1, undergo the second stage of the meiosis, that is the meiosis-2, and through an equational division like that of mitosis, the result is the four types of haploid cells or haploid gametes, as you can see in this sketch. Now we know that meiosis is completed in two sequential cycles, that is meiosis-1 and meiosis-2. Now first of all we will discuss about the meiosis-1. This division, meiosis-1, results in the reduction of chromosome number to half, thus is known as heterotypic or reductional division. This division or the karyokinesis of the meiosis-1, that is the nuclear division of meiosis-1, consists of four phases. That is, number first is prophase 1, prophase, then metaphase first, then anaphase first, then telophase first. Now we will discuss about the prophase first. As you can see in this sketch, this prophase first is divided into 5 substages, that is leptotene, zygotene, pachytene, diplotene, and the 5th stage, that is diakinesis. So far the first stage is concerned, that is the leptotene. During this stage, condensation and coiling of chromatin fibers lead to formation of distinct chromosomes which appear as fine single threads of a bead. As they contract and become thicker, their dual nature is seen, as you can see in this sketch. Then after the leptotene, the next stage is the zygotene. During this stage, as you can see in this sketch, the homologous chromosomes start pairing, which is also known as synapsis. It is accompanied by the formation of synaptonemal complex, and the chromosomes occur as bivalents, paired. Now after this there is the third stage, that is the pachytene. During this stage, the two sister chromatids of each chromosome become visible and bivalent, as you can see in this. They are also known as tetrads. Now recombination nodules the sites of crossing over appear. Crossing over is catalyzed by enzymes like recombinase. Recombinase is employed here in lectin for crossing over. Now after this, the fourth stage of prophase 1 is known as diplotin. This diplotin stage is marked by the dissolution of synaptonemal complex and the homologous chromosomes start separating from each other except at the sites of crossing over, as you can see at these sites, and these sites are known as chiasmata. Now after the diplotin, the fifth and the last stage of prophase first is what we call as diakinesis. As you can see in this sketch, this chiasmata moves to the tip, or it is also known as terminalization, and may slip off or remain at the tips. The diakinesis represents the transition to the metaphase first. Then this completes these five stages. With these five stages, the prophase first is completed, and then we come to the metaphase first. During this metaphase first, the bivalents, as you can see in the sketch, which are also known as tetrads, are arranged at the equator of the spindle. The centromeres are aligned in two rows during this metaphase first. Then we have the anaphase first. During this anaphase first stage, the members of every homologous pair of chromosomes start separating from each other and start moving to the opposite poles of the spindle. The centromeres, as you can see, do not split at this stage, and the number of chromosomes becomes half the number of chromosomes of the parent cell. Then we have the telophase first. During this stage, telophase first, the chromosomes have reached the poles, as you can see here, to opposite poles, and a nuclear membrane starts to form around the chromosome cluster at each pole, giving rise to two daughter nuclei. Now this completes the first stage of the meiosis, that is the meiosis first. Now in our next topic, we will discuss about the meiosis second. Until then, goodbye.